All right, Owls, this is Mr. O with your first set of flip notes that we will do this year. Flip notes are typically done at home. This first set we're going to do in class just so you get comfortable with what the process is. And if you have any questions, I'm there to answer them today. Um, Always with the flip notes, we will have your binder number on the first slide, and we will do that in class, but you'll come back and put that page when it's completed in your binder, and you'll use these notes as support for when we do activities, labs, projects, studying for tests, etc. Things to remember with flip notes. First of all is you complete the note template that goes along with the video. I gave you this note template. You have that. As you go along with the video, it goes in order, and you will complete the template as you find things in the video that match the template. You can pause, you can go back, and you can rewatch this video at your own pace. This is the beauty of flip notes. If you work fast, you can work faster. If you want to take your time, you're more than welcome to do that, and no one in class knows because you are at home doing this. If you have any questions about the content that we are discussing in flip notes, please jot them down so you can ask them when we get back to class. The idea of flip notes is that I don't give you the initial notes, but we dig deeper in class with activities. So if you have questions about the content that you talked about, write them down. I'll be happy to ask and answer them when we get back to class. And be sure to watch and listen to the entire video. There are great explanations and examples that will not be in your note template, but they'll help you understand what we're learning better. This is why flip videos are nice, because you can always go back and rewatch if you are confused or certain things are giving you trouble. And my explanations and examples could help better understand the material. Now, let's get started with the notes. The first thing on your notes you see is the title Different Methods. And we're going to talk about two different methods used by scientists. And the first of which would be the scientific method. So we have a poster here that you typically would see in a science class, and then you have another poster here that you'd also typically see in the science class. So the scientific method is the first of two different methods. The two posters are very similar, except for the first part. It says one says purpose and the other one says question. Those actually mean the same thing because your question should be your purpose. And then we have the engineering design process. And this is a little bit different because of the fact that it's not a linear method. It actually can start anywhere depending on your task. So the two different methods used are the scientific method and the engineering design process. Let's focus on the scientific method. Obviously, this is what the flip notes are based on. This is the first thing that we're going to complete in class. If you look at your template, you have the 6 general steps of the scientific method, and I will give you definitions for each of those 6, the first of which is question. You have to create a testable or experimental question. That is your purpose in your experiment. There is no starting the scientific method unless you understand why and what you are doing, and the question will do that. Then we have hypothesis. I will argue that you will never make a hypothesis until late high school, maybe college, if ever, in your science career. We will usually make educated predictions, so that will be my definition for hypothesis because it's more applicable to 6th grade. Hypothesis is you're making a prediction of what's going to happen. The experiment is the fun part. You design an experiment, you have control and experimental groups, meaning the control is what you compare to. And in the experiment, your goal is to collect data. And we talked about in class that you use your 5 senses to collect data. And the data is simply information that you'll use to make accurate questions and answers from your experiment. The next is analysis. So you take the data that's collected, you compare one to the other. And this is where you're starting to learn, am I right? Am I wrong? What happened? What do the results tell me? That will all be done in the analysis part. Next is the conclusion. Typically, conclusions come at the end, and this is where you combine your prediction with your data analysis, just like we did with the ink lab. I asked you to look at your prediction, and I asked if you were correct or not, and I said use evidence from the lab, and that data analysis is the evidence from the lab. So in every conclusion, you should be talking about your prediction and what happened in the experiment, and you should also be providing evidence. Last but not least, you should be communicating, which means sharing out the information that you got. This could be in the form of a lab report, this could be in the form of a vlog or a blog or a post, or even just discussing with classmates. So those are the 6 steps of the scientific method. You can find the scientific method that is done with different words, but in general, these are the 6 steps that they are all work along with. Now, on the back side of your footnotes, we have designing an experiment, groups, variables, types of data. First, we're going to talk about 2 different groups. The first group is called the control group. This is the group that does not get tested against the variables in an experiment. Lots of times when doctors issue new prescription drugs to the public, they will test on people if these actually work. The control group gets a fake pill, and the experimental group gets the real pill. So what this is they're actually taking these 2 groups so they can compare them against each other. If you do not have a control group, you cannot know if your experimental group works. So your experimental group is the group tested against the variables, and you can only test one variable at a time, because if you did not just test one variable at a time, you would not know what is changing and be accurate on that in your data analysis part. So the 2 different groups, and this is based in your experiment, and you can't make an experiment without these 2 groups. Now we're going to talk about 3 types of variables. Now these will come up throughout the year. The first of the variables is an independent variable. These are variables that are changed in an experiment. I underline the word I in the next statement. It says what I changed so I can test my hypothesis. Let's use the ink lab as a description. We used different markers in the ink lab. That was my independent variable. I changed the markers so we can test each marker against another. And I only did one type of marker on each strip, so we knew that that marker was that result. The second is dependent variables, the changes that come from the independent. So the independent variables will lead to your dependent, and you see I underline the word D for data in the next statement. It's the data you collect. These are your results. In the ink lab is the different ways it's spread on the paper. And last but not least, you have a control variable, and this will be based on the format of the experiment. Let's use that ink lab one more time to explain what happened. I used the same coffee filter for all strips. We put all the strips in the same water in the same time. The dots were the same distance from the bottom, and I used the same amount of ink on each of the strips, so I was able to know that that experiment was controlled, and I can take data from that and be accurate on my analysis. So the ink lab, those are the 3 types of variables and how I use them. And last but not least, you have 2 types of data, or data, however you want to say it. Tomato, tomato, as I would say. You have quantitative data, and that's data that can be expressed as a number or quantified. And you usually find this through measuring of some sort. It could be weight, mass, volume, distance, length, all those fun things. Anything that's a number would be known as quantitative. And I remember that because quan has an N in there, an N with number. And the other type of data is qualitative data, and this is data collected using the 5 senses. Each of these data are important and valuable, but sometimes you do a lab that only uses quantitative, and sometimes you do a lab that only uses qualitative, or sometimes you have a lab that has both. But you need to know that there's 2 different types of data in an experiment in a lab. That is the end of Flip Notes. Remember, if you have any questions, please write them down, and you can ask them when we have a discussion about them. And at this point, your notes should be completed. Hope you enjoyed Flip Notes. We will be using these throughout the year.